Hi everyone, welcome back. Today we're diving into CovalentLab, a new platform for rational covalent drug design. Over the past decade, covalent drugs have reemerged as a powerful modality in drug discovery. By binding irreversibly or partially irreversibly to their protein targets, they achieve long residence times, strong potency, and in many cases the ability to overcome resistance mutations. Yet there are also limitations with covalent drugs like possible off-target interactions and so on. Over 100 covalent drugs have been approved by the FDA in these past 50 years. Most current covalent design still revolves around cysteine and serine, even though many pockets contain other nucleophilic residues that could, in principle, be targeted. This paper introduces CovalentLab, an interactive AI-powered platform that enables rational, one-shot design of covalent ligands. CovalentLab can work in two ways. You can start from a known ligand that already binds your protein target or from a reactive warhead you'd like to use or you have designed. In both cases, it predicts which residues could react and how to attach the warhead to generate covalent candidates in 3D. A central result motivating CovalentLab is a structural analysis of PDB-bind complexes. The authors found that over 95% of binding pockets contain at least one nucleophilic residue within 6 angstroms of the ligand. That means many reversible binder could, in principle, be converted into a covalent inhibitor. However, not all residues are equally reactive. But how to identify which ones are actually good covalent target sites? To address this, the authors fine-tuned a pre-trained ESM2 protein language model using LoRa, creating a deep learning classifier that predicts the covalent reactivity of 9 amino acids ranging from common covalent targets like cysteine or serine to rarer ones such as tyrosine or histidine. CovalentLab unifies two complementary covalent design strategies. You can use the covalent molecule design approach. Here you start from a protein-ligand complex in 3D. The ligand binding pocket is analyzed and the model predicts covalent binding sites. It identifies atoms on the ligand that could be modified. Then, users can choose from a diverse library of covalent warheads and explore various modification strategies. Or you can use the module named Covalent Warhead Application. In this case, you begin with an electrophile you want to use. The platform screens the protein data bank to find protein-ligand complexes suitable for warhead conjugation. Several strategies can be selected, such deciding the amino acid type on the protein target. In both cases, the output includes recommended amino acid residues, generated covalent molecules, docking poses, and downloadable structures. This gives users a complete end-to-end workflow for covalent ligand design. To demonstrate scalability, the authors have developed and integrated a comprehensive online resource centered around a curated dataset of more than 100,000 covalent small molecules, meticulously designed across 95 protein targets to facilitate the discovery and rational design of covalent inhibitors. This is the library section of the platform. Each molecule is annotated with its parent ligand, targeted residue and mechanism, 3D docking pose, drug likeness metrics like Lipinski, QED, assay score, and interactive 3D visualization. The chemical space of this library goes far beyond existing covalent inhibitor databases, offering a rich resource for downstream screening and possibly new machine learning applications. The platform was validated experimentally on two important protein targets, tropomyosin receptor kinase A and glutamate C, to try to overcome resistance mutations or to design a covalent inhibitor binding to an allosteric pocket. These results highlight that CovalentLab doesn't just generate theoretical ideas, it produces experimentally validated covalent hits. To summarize, most protein pockets have nucleophilic residues potentially suitable for covalent targeting. CovalentLab provides a unified AI-driven workflow for identifying those residues and designing covalent ligands. It supports both 3D ligand-based and warhead-based design, including custom warheads. More than 100,000 covalent molecules were generated across diverse targets. Experimental validation on 2 protein targets shows that the platform can produce real, biologically active covalent inhibitors. Overall, CovalentLab represents an important step toward data-driven, rational covalent drug design, moving beyond cysteine or serine targeting and expanding what is druggable by covalent chemistry. I hope this video will be useful for your project, and until next time, take care.